Welcome students. In this video series, I will going to solve numerical problems related to biology that appeared in previous year IIT JAM examination. In this video, I will discuss the questions that appeared in 2016. So without wasting time, let's see how to solve those problems. The first question is from the topic amino acid. Here the sequence of a short peptide is provided in 3-letter abbreviation and you have to calculate the net charge of the peptide at pH 7. When this type of problem appeared, your first task is to find out the charge of each amino acid in this way. Here aspartate and lysine are charged amino acid and cancel each other, so the net charge will be 0. In this problem, the length of a bDNA is provided and you have to calculate the number of turns present. We know that according to Watson and Crick bDNA model, one complete turn is 34 angstrom or 3.4 nanometer. So in 152 nanometer, there will be 44.70 turns. In this problem, a population is in Hardy-Weinberg equilibrium for a gene. There are two alleles, that is capital A and small a. The gene frequency of capital A is 0.7. You have to calculate the genotype frequency of heterozygous. We know that according to Hardy-Weinberg, when mating is random in a large population with no disruptive circumstances, the law predicts that both genotype and allele frequencies will remain constant because they are in equilibrium. So capital A plus small a is equals to 1. Then capital A is 0.7. So small a will be 0.3. Again, we know that this relationship is also 1. So using this, I have calculated the genotype frequency of heterozygous and it is 0.42. In this problem, a receptor binds to its ligand and the dissociation constant is 10 to the power minus 8. We have to calculate the ligand concentration when 10% receptors are occupied by the ligand. We know that the binding equilibrium called capital Y is equal to the binding site occupied divided by the total binding sites. So here capital Y is equal to 10 divided by 100 or 0.1. Now we will calculate the ligand concentration using this equation. Here L is the concentration of ligand and Kd is the dissociation constant. Here it is 10 to the power minus 8. When we solve it, the concentration of L is 10 to the power minus 9. In the next problem, an enzyme converts 4 into 10 to the power minus 4 molar substrate into product at a rate of 20 micromolar per minute. The Km is provided and you have to calculate the value of Vmax. To solve this type of problem, we will use Michaelis-Menten rate equation. Here substrate concentration S is 4 into 10 to the power minus 4 molar. Initial velocity V0 is 20 micromolar per minute. Michaelis-Menten constant Km is 2 into 10 to the power minus 4 molar. We will put all those values in this equation and solve for Vmax. Here Vmax is 30 micromolar per minute. In this problem, 10 to the power 6 bacterial cells were there and doubling time is 60 minutes. And you have to calculate the number of bacteria will be present in the medium after 10 hours. First, we will convert the doubling time, that is small g, into minute and it is 600 minutes. Now we will calculate in 10 hours how many generations will pass. We know that small g is t by n, where n is the number of generation and t is the time. So n is 10. Again we know that nt is n0 into 2 to the power n. Here n0 is the initial bacterial number, nt is the final bacterial number. When we solve it, the value of nt becomes 1024 into 10 to the power 6. Now this peptide was sequentially treated with dithiothiotol, cyanogen bromide and trypsin. We have to calculate the number of fragments will be generated after the treatment. We know that dithiothiotol is used to break disulfide bonds if present. The cyanogen bromide digests peptide after methionine residue and trypsin breaks if lysine or arginine is present. So let's watch carefully. Here no disulfide bonds are present. So after dithiothiotol treatment, no fragments will be generated. When cyanogen bromide is used, it will cut here and here. Then upon trypsin treatment, the peptide will break here and here. So total number of fragments will be 5. Here in this problem, colored and round seed is dominant and colorless and shrunken seed is recessive. Pure double dominant variety is test cross with F1 generation plant and the result are provided in this figure. You have to calculate the distance between the seed color and seed shape genes. To solve this type of problems, our first task is to find out the total number of progeny, that is 800, and then identify the parental type and recombinant type. Here colored round seeds, that is 380, and colorless shrunken seed is 396 are parental type. Then we will find out the recombination frequency using this equation. So the distance between the two genes are 3 centimorgan. In the last question, a globular protein contains 50 amino acids and out of 50 residues, 24 residues are in alpha helix and 26 residues adopt beta sheet. You have to calculate the length of the protein in nanometer when it is fully extended. We know that alpha helix per turn there are 3.6 residue and pitch is 5.4 angstrom. So 24 residues will be 3.6 nanometer. And in beta sheet, the distance between two residues is 7 angstrom. So 26 residues is 91 angstrom or 9.1 nanometer. So the total length will be 3.6 plus 9.1 or 12.7 nanometer. I hope you will find this video helpful for your exam preparation. So press the like button and make your comments here. Subscribe my channel for more videos like this and press the bell icon for regular updates. The subtitle of this video is also uploaded. You can visit the community section for more questions like this, and thanks for watching.